Hello, I'm Jeff Sawyer, CCI's Director of Employer and Professional Network Development. This Future Forward segment features Heidi Rehm, Chief Genomics Officer for the Center for Genomics Medicine and Department of Medicine at Massachusetts General Hospital. She's also the Medical Director of the Clinical Research Sequencing Platform at the Broad Institute and a Professor of Pathology at Harvard Medical School. Heidi has over 25 years of experience in the genomics field, from her PhD program in genetics at Harvard, which she began directly from her Middlebury graduation, to her present roles. Heidi graduated from Middlebury in 1993 with a degree in Molecular Biology and Biochemistry. Heidi is joining us to share her perspectives on the field of clinical genomics, including a brief overview of the genomics field, some of the challenges and opportunities influencing the future of this field, and her career advice for students to position themselves to enter this field in the future. Heidi, thank you for joining us.
It's my pleasure to be here.
Just to set the stage for our conversation, can you give us an overview of the field of genomics?
Sure. When we talk about genomics, first to contrast with genetics, some people say, what's the difference between genetics and genomics? When we talk about genetics, we're usually talking about focusing on particular genes and their functions or their roles in disease, perhaps the CFTR gene that's implicated in cystic fibrosis, so sort of one or a few genes that we might study. When we move to the field of genomics, it's really an approach where we look at the entire human genome of an individual or of a population and try to understand what roles different genes or combinations of genes might play in a human phenotype or a biological function. That's really when we think about genomics, we're really looking at the whole set of genes that make up a human individual.
Can you tell us a little bit about the main contributors to the work in the field?
So there's lots of people that work in the field of genomics, and there's both researchers who are studying individual genes, collections of genes, and their role in certain diseases. You might have a clinical researcher with expertise in a particular disease area, cardiovascular disease, hearing loss, different areas where researchers are studying the functions of those genes and how they lead to disruption and human disease. You also have clinicians who practice in different fields of medicine, including a medical geneticist who might see children or even adults with certain genetic disorders. You also have genetic counselors. This is individuals who might get a 2-year degree, really focused on allowing them to meet with patients and counsel them on how their genetics might impact their health and contribute to disease that may be inherited or passed on to a child. And so really helping understand those contributions. And then we also have individuals like myself who work in clinical laboratories that oversee genetic testing of individuals and how different variants in their DNA might contribute to disease that they might manifest. And so there's variant scientists or clinical laboratory directors who oversee and participate in the laboratory testing process.
Let's stay with that for a little bit. Can you tell us a little bit more about the work that you do at Mass General and Broad Institute?
Sure. So many years ago, in about 2002, I was hired to build a new genetic testing lab. And that laboratory spanned Mass General Hospital and Brigham and Women's Hospital. And we began developing genetic tests in disease areas that never had genetic testing. We launched the first genetic test for a disease called hypertrophic cardiomyopathy, which is a heart condition. And some of you might have heard something about this because you often hear about it when a professional athlete dies suddenly. Hank Gathers, Reggie Lewis, they died of a disease called hypertrophic cardiomyopathy. It's also dominant, so it can be passed on to other individuals in their family. And we do genetic testing so that we can figure out the cause of their disease and also test other family members who might be at risk. So my role has over the years has been directing a laboratory that has developed these types of new tests. We also launched the first genetic test for lung cancer treatment to identify certain variants in a gene called EGFR, for example. There's others that contribute now that help dictate which drugs to treat a patient with lung cancer with. So these are different genetic tests that my laboratory over the years have been, running and being able to diagnose patients with different genetic variants that contribute to their disease or how to treat them. And so I ran a lab at Mass General Brigham for about 17 years. In the last 5 years or so, 5 or 6 years, I've also been the medical director of the Broad Institute's clinical laboratory. And we offer many different genetic tests, particularly genomic tests like whole exome sequencing, whole genome sequencing, partnering with other laboratories to offer those technologies at scale. We can, sequence thousands of genomes a week, genomes every day, to enable broader genetic testing where we can interrogate every gene in the genome when we're trying to understand why a patient might have a certain genetic disorder.
Interesting. Can you give us a sense of the scale of this particular field within the healthcare profession?
Right. So, there's different aspects of scale. So today, in clinical genetic testing, adoption of genetic tests has happened heavily in certain disciplines and yet to be fully implemented in other disciplines, and it varies a bit. Probably the highest volume testing in our hospital today and in many hospitals around the world is in hereditary cancer testing. And there's a very strong appreciation for the role of genetics in leading to development of cancer at a higher rate than we see in the average population. So not every cancer has a genetic basis to it or is hereditary, but some cancers are. And so when a patient develops cancer, particularly at a young age, or particularly when multiple members of the family develop cancer, that's really the times when you want to test different genes to figure out if there's a hereditary component. And then you can test other family members for that known variant and be able to predict who's at risk, intervene early, and be able to prevent, the serious outcomes of someone developing cancer and not being detected until late stages, which we want to try to avoid. So, certain disciplines like cancer is an area where there's been lots of genetic testing that's been utilized in medicine. In pediatric care and neurodevelopmental disorders, intellectual disability, seizures, these are other areas where genetic testing is widely utilized in the diagnosis and care of patients. Certain cardiac disorders, I mentioned hypertrophic cardiomyopathy, but also arrhythmias, familial hypercholesterolemia, when you have very high levels of cholesterol, that can often be due to a genetic basis. And so in cardiac disease, genetic testing is fairly widely used. There's other disciplines where it's still developing its applications. I think kidney disease testing has been a little later to sort of jump on board, but we've been rolling that out here at Mass General Hospital, and it's been very effective at diagnosing patients for whom we didn't have an explanation for why they had disease. I think another area where genetics is being widely used at scale is in research. So I talked about the clinical application, but there are increasing genetic studies being done where populations of individuals at large scale are being enrolled, either through their healthcare system or through independent research programs. And those individuals' DNA or blood specimens are being collected, and then we can sequence their DNA from their blood, and large sets of data are being aggregated. For example, I'm involved in the All of Us research program. That's a research program funded by the NIH here in the US. And we are aiming to enroll at least 1 million individuals And at the Broad Institute, we are one of 3 centers where we are sequencing the genomes of this large cohort. And we've already generated 60,000 whole genome sequences here at the Broad Institute. For individuals and all of us, will have over 100,000 genomes sequenced by the end of the year for this program, and we'll continue on sequencing until we reach at least 1 million individuals in that cohort.